we're going to change now from processing side of things, and we're really going to say, okay, tell us about what this whole area of protease activity within the seed itself is. And Dr. Lisa Weaver is going to lead that discussion. She's trained as a biochemist and has for a considerable amount of time has worked with USB through Smithbucklin, leading the agronomic side of things with it, working with geneticists and also into measurement of nutrients in that area. So
we're going to switch over from plants made out of concrete and steel to plants that are green. So thanks for the introduction. As you know, I'm not a poultry scientist, so I'm going to be talking about this strictly from the plant side. So let's see if we can get on to the next slide. Yay, it works. Can't hear? Okay. So before we talk about protease inhibitors, let's talk for a couple of minutes about plant proteases. They have a number of different roles in plant growth and development. So they're involved in fertility, they're involved in germination, they're involved in organelle function, they're involved in plant defense responses through apoptosis, general cell cleanup, so degradation, turnover of protein. In model plants, there's more than 700 genes that have been described that are proteases, and that's really a lot of genes. The physiological function of these is not strictly known. There's been some attempts to do, systematic functional genomics, where you either overexpress or knock out genes. And maybe 40, 50 phenotypes have been noted by doing that. There's a lot of, when you do that, there's some that don't show any phenotype changes at all. So there's some redundancy there. From the nucleic acid sequences, the major classes of proteases are all present. Threonine proteases, which are a very tiny fraction in plants. Metalloproteases, which are in a minority, and then the majority are serine, cysteine, and aspartate proteases. Okay, so here's some pretty ribbon structures of plant proteases. There's an example of a cysteine protease from pinwheel flower, which is a tropical plant that's a medicinal herb. I have no idea why somebody wanted to crystallize this protein, but they did. It doesn't have any physiological function that we know of. There's the tomato serine protease, which is subtlysin-like. This is a nice example because it's a homodimer, and you can see the two subunits with the active site that lies in the cleft between them. It's probably involved in defense response. And then the barley aspartate protease that's found in the vacuole, probably involved in protein turnover. All right. Okay, so let's talk a little bit more about plants, but also soybeans. So there's each one of these types of proteases, the cysteine proteases, aspartate proteases, and the serine proteases. They have proteins that are also inhibitors of them. So the cysteine proteases have the cystatins. The aspartate protease has aspartate protease inhibitors. It's a good name, a little long, but it's good. And then the serine proteases have the serpins, which are proteins that inhibit serine proteases. Although there are there's some overlap there, not in soybean, but in other plants that have been described that will also inhibit cysteine proteases, so they're known as promiscuous. So let's narrow down on the serpins superfamily. It's really widespread in plants. Pretty much they occur broadly in monocots, in dicots, in mosses, in algae. Pretty much if you have something that's green, it is going to have a serpin in it. They've been better characterized from some of the row crops and from model plants like Arabidopsis. if you think back to all the different kinds of genes that are present in plants for proteases, the serine proteases are a significant amount of those, and that could account for why these serine protease inhibitors are also pretty widespread in plants. Okay, so the serpins are ubiquitous also in plant organs and tissues, because that's the next question I always ask. Okay, it's in the plant, where is it in the plant? A little bit different between monocots and dicots. So what's shown here in the middle of that slide is lovely structure for the Kunitz trypsin inhibitor, KTI, or the Bowman-Burke inhibitor, which is below it, the BBI. And that just as examples of serpins. And in monocots, they're found in all the vegetative tissues, so the leaves and the roots, and in the seeds, and they're found in dicots, in the leaves and the roots and the seeds. Surprisingly, the richest source in monocots is actually in the seed. And for dicots, it's predominantly in the vegetative tissue. So now let me talk about that a little bit, because you guys are nutritionists. So the cereal grains are, as you know, the starchy endosperm is starch. It doesn't have a lot of protein. So even though it's a quote-unquote rich source, it doesn't mean there's a great deal of it necessarily. It's just rich compared to the other parts of the monocot plant. Okay, so soybean serpins, the Kunitz and Bowman-Burke trypsin inhibitors, are what's known as suicide inhibitors. So there's a business end of the molecule here that interacts with the proteases, and they're called suicide inhibitors because as they go to interact with the protease, they kill themselves. They are actually acted on by the protease, and then they become covalently bound to the protease, and then there's nothing left to do with that except send it off for protein turnover. So they inhibit their targets in a 1-to-1 molar ratio. And you can see here there's crystal structures of the pig trypsin with KTI and cow chymotrypsin with the BBI. So what are these things doing in plants? So KTI and BBI are protective for plants. They're made in response to stress. That can be biotic stress or that can be abiotic stress. So while you guys are worrying about, chickens eating soybean meal maybe, the plants are worried about what is eating me. And so they're worried about primarily insects, but it can also be animals, rodents, or birds, or other things that you find out in the wild. And so these protease inhibitors will impact the proteases that are found in the guts of insects and other animals. For abiotic stress, there's been some indication that they may provide a protective function during things like drought stress and water stress or flooding stress, where those proteins, it's not clear exactly how they're working, but it's suggested that they might be helping to keep endogenous proteases from turning over proteins during a stress response. One more thing I wanted to say about seeds. So the trypsin inhibitors accumulate in seeds at the same time as the seed storage proteins. And that may be just because that's when they're all laid down. It may be because they might be providing a protective function for the seeds not to be eaten by animals. And it could also be that they're ensuring that during the germination process, because seeds are metabolically active all through their quiescent stage, and although at a really low level, and then that continues and ramps up during germination. And so things in germination have to go during a certain set program, and these inhibitors may be helping to protect seed storage proteins or proteins from being turned over until it's time to do that. Okay, so anything that denatures a protein will get rid of your trypsin inhibitor activities. for these protein trypsin inhibitors. So any kind of physical process like heat, ultrasound, extrusion, if you can get enough shear and enough flash heating, will work. Reducing agents, acid or base, and then biological processes. So germination, you can get rid of trypsin inhibitors by germinating seeds or by fermentation, because during the fermentation process, there's other proteases that will chew up those proteins. So this slide is here to remind me to talk about engineering trypsin inhibitors. So we have that ability to do that in plants. People have actually overexpressed trypsin inhibitors, and that's really more for trying to protect the plant against an insect or against a stress response. It's been shown that you can overexpress KTI and BBI in soybean seeds, and you can improve resistance to corn earworm, which apparently also eats soybeans. But in your case, you're probably most interested in trying to get rid of trypsin inhibitors in the seed. So it's estimated there's around 10 or 11 KTI, 10 genes for KTI and about 11 for BBI in soybeans. And a lot of this work has been done by Krishnan, and he's in the audience here, and he'll be talking in a few minutes. But natural variants without KTI activity have been characterized. And Hari found that, the more you try to knock out KTI, the more BBI you get. So the plant compensates for trying to get rid of KTI by increasing another trypsin inhibitor. There have been some reports recently of lines edited to contain less of the Bowman-Burke inhibitor, and those have an 80% decrease in the chymotrypsin activity. So now the perfect storm is, to bring those two things together. Will the plant let us do that? We don't know. Time will tell. So take-home messages. Proteases are really important to plant development, and all the roles of proteases in plants really haven't been fully elucidated. Plants make inhibitors of proteases. The aspartate, the cystatins, and the serpins. The serine protease inhibitors, the serpins, are ubiquitous, and they're found in most tissues and organs. Two classes of serpins, KTI and BBI, are suicide inhibitors. They probably have protective functions in plants, and so they're there for a reason. The plants need them, especially in the vegetative tissues. Processes that denature proteins should get rid of these things. And progress has been made towards seeds without trypsin inhibitor activity. And I can take any questions.